Adaptive immunity includes cell-mediated immunity and humoral immunity. Right now we will be talking about cell-mediated immunity, which is also called T-cell-mediated immunity because it involves T cells or T lymphocytes. One type of T lymphocyte is the T helper lymphocytes. There are two types of T helper lymphocytes. There is T helper 1s and T helper 2s. Both T helper 1s and T helper 2s have a specific TCR molecule, T cell receptor, that binds to a specific antigen. They also both express the CD4 molecule. Because of this, they can only bind and recognize antigens that are presented on the MHC2 complex. The MHC2 complex is located on antigen-presenting cells, some antigen-presenting cells. The only ones it's located on are the dendritic cells, the neutrophils, and the macrophages. T helper 1 cells help control bacteria infection, so the antigen-presenting cell is likely a bacteria-infected cell. In other words, it's also here. So the antigen-presenting cell, either a dendritic cell, neutrophil, or macrophage, has a MHC2 complex, and this presents an antigen. And since it's on an MHC2 complex, the T helper cells can recognize it with their TCR specific receptor, and then it's also involved the CD4 molecule. And a way to remember this is that T helper has an H, and if you invert an H, there's a 4. And then you can remember that CD4 matches with MHC2 because 4 times 2 is 8. And we'll see later that the cytotoxic T cells will also give a product of 8. The product of 8 doesn't mean anything, but it's a good memory trick. So, T helper 1s also help activate the cytotoxic T lymphocytes. These cytotoxic T lymphocytes are the primary cells involved in cell-mediated immunity. It's called cell-mediated immunity because the two cells come face to face and the cytotoxic T cells kill the infected antigen-presenting cell. The cytotoxic T cells have a specific TCR as well, but they have a CD8 molecule, which requires the antigen to be presented on an MHC1 complex. And this MHC1 complex is found on all nucleated body cells, so it's on lymphocytes, just every body cell, except not red blood cells because they don't have a nucleus. And cytotoxic T lymphocytes help kill viruses, tumor cells, transplant cells, and fungal cells, but they do it specifically. Later, if you want to compare it to the natural killer cells, they also kill viruses and tumor cells, but natural killer cells do it non-specifically since they're part of the innate immune system. But natural killer cells are not part of the cell-mediated immunity. Yes, so then that can be illustrated here. So there's a antigen presenting cell that could be any infected nucleated body cell, and it presents an antigen on an MHC1 molecule, and a cytotoxic CD8 positive T lymphocyte with a specific TCR that matches this antigen and that has a CD8 molecule can bind to this complex and recognize it and activate it with the help of T1. So activation with cytotoxic T cells involves T1 and the binding. And you can remember that cytotoxic T lymphocytes are CD8 positive, and they match with MHC1 because 8 times 1 is 8. So both matches produce a product of 8. T helper cells are CD4 positive, and they recognize MHC2. 4 times 2 is 8. And then cytotoxic T lymphocytes are CD8 positive, and they recognize MHC1. So that's 8 times 1 is 8. Okay, and then now we're going to move on to humoral immunity. And T helper type 2 cells, they help activate B cells, which ultimately help produce the humoral immunity. Humoral immunity is adaptive immunity that involves antibodies. So this is more of an extracellular thing, because humoral refers to the humors, which are the fluid in your body, whereas T cells dealt with intracellular pathogens that were inside cells, but these are antigens and bacteria floating in the humors of the body extracellularly. So here's a naive B lymphocyte, and they have a specific B cell receptor, BCR, that binds and recognizes a specific antigen. And once this occurs, this selects this B cell, since it bound to the correct antigen, via clonal selection. And then via clonal expansion, it will proliferate and multiply into identical cells. And then these cells can differentiate into plasma cells and memory cells. The effector plasma cells are the ones that secrete the antibodies, which creates the humoral immunity. And then the memory cells, the memory B lymphocytes, they contribute to immunological memory. And this is why there's a difference between the primary response and the secondary response. So primary response is the first exposure to an antigen. And here it takes a while for antibodies to be produced, because B cell activation takes 4 to 5 days. But in the second exposure or subsequent exposures to the same antigen, there are memory cells, so there's a faster and stronger antigen or antibody production. And then, so how do antibodies help the immune system? They have some different actions. So one is they can neutralize a antigen. So they bind to the antigen, which prevents it from interacting with other cells here. Or there's agglutination, which they form clumps of antibody complexes, because antibodies have 2 antigen binding sites. And then there's opsonization, which promotes phagocytosis, because, well, you don't really need to know this, but phagocytes can recognize this end portion of the antibody, the FC region, the constant region. And then it can also activate complement, which can insert a membrane attack complex, MAC, and then that causes cell lysis, so the infected cell will die. And then there are 5 different types of immunoglobulins or antibodies. IgG is the most abundant, but it's the smallest in size. And for that reason, since it's so small, it's the only one that can cross the placenta. And then IgA is the primary immunoglobulin found in breast milk. IgG is also in breast milk, but IgA is the primary one. I like to remember this because there's breast milk, and then I think of like 8 cup bra sizes, and yeah, that helps me remember it. And so IgA is passive immunity, or IgA in breast milk passed from the mother to the infant is a form of passive adaptive immunity, because Passive adaptive immunity is when preformed antibodies are passed to a person, but no actual immune response was created. Adaptive immunity is when your body is presented with the antigen or the pathogen in the form of real infection or vaccine. And then IgM is first to proliferate in the primary response, and then IgG proliferates the strongest in the secondary response. And then IgD is involved in B lymphocyte activation. And IgE is the deadliest, so you'd want it in the smallest amount. So yeah, IgE is the deadliest because it can kill you via anaphylactic shock. We'll get into that when we talk about type 1 hypersensitivity, and is located on mast cells and basophils.